Hello everyone. Let's start with HIV replication cycle. Now HIV is a retrovirus. Retro meaning it behaves backwards from the original way. So now what is the original way? So the original way is central dogma. In the central dogma, what happens is there is a DNA. DNA is the genetic material of all cells. This DNA gets transcribed into mRNA. And this mRNA gets translated into proteins. And these proteins are used for various purposes. And this happens in all living cells, but there are always exceptions. So, the retrovirus is one of them. In a retrovirus, RNA is the genetic material. And this RNA gets converted into DNA and then the proteins are formed. Okay? Right. Now, let's give a quick recap to structure of HIV. I'll label it GP120. Now this GP is the glycoprotein and 120 is the molecular weight of the protein, right? Then this one is envelope or lipid membrane. This is protein coat. This is capsid. This is single-stranded RNAs. And the red dots which you can see are the enzymes. Okay, let me label them also. This is reverse transcriptase enzyme. This is integrase enzyme. And finally, the third one is protease enzyme. All of the functions we are going to see soon enough, right? Now, remember this GP120? It is the most important part. How come? Well, this GP120 is going to recognize receptor on the surface of host cells, right? Now, which receptor it's going to recognize? It is going to recognize CD4 receptor. Right? Now, is the CD4 present on all cells? No, it's present on certain immune cells. Right? There are many immune cells in our body, but CD4 receptors are present on T cells, monocytes, and macrophages. Right? Now, what is the function of CD4? Now, this CD4 is used for signaling other parts of immune system the presence of antigens or pathogens, right? Now this virus is very smart. It is directly going to bind with CD4, right? It is directly going to be binded with CD4. Okay. Now let's see the cycle, right? Now if you can see here, then this portion is the nucleus and this portion is the membrane, right? Okay. Now virus is there already. Virus is there, right? Here virus is there. Now this virus is going to bind with CD4 receptor. So there should be a CD4 receptor. Here it is, right? This is CD4 receptor. Let me just label it CD4 and here this GP120, right? Now as soon as this GP120 binds with CD4, the binding has occurred. And plus to support it, there is also co-supporter, right? It is known as CCR5. Once the binding occurs, what's going to happen? The virus needs to enter inside. So how is this possible? By the fusion of the membrane of host cell and the virus, right? So for the fusion, right, here the fusion occurs of this host cell membrane and virus membrane. Okay. Once the fusion occurs, what's going to happen? Once the fusion occurs, RNA is going to enter inside the cell. Right. So RNA enters inside the cell. Right. RNA enters inside the cell. Now from here we'll focus on only single RNA. Right? Both the RNAs will work in the same manner. So we'll focus on single RNA. Okay, so this is RNA and this is the enzyme present on it. Okay, so this enzyme is reverse transcriptase. Now this enzyme will get activated, and this enzyme, what's this enzyme is going to do? It's going to convert single-stranded RNA into double-stranded DNA. Now this DNA is formed. So after the formation of HIV DNA, it is going to enter into the nucleus of the host cell. Right? Now we know that inside the nucleus already host DNA is present. Right? But what is the main task here? The virus needs to enter inside the host DNA. Right? So this is here. This is here virus double-stranded DNA, right? Now what's going to happen? Enzyme integrase is going to get active. Okay, this integrase is again from HIV, right? So this enzyme is going to get active, and what's going to do? It's going to incorporate this double-stranded DNA inside the host DNA, right? So supposing now what will be the structure? Okay, this is the host DNA. Then this integrase is going to incorporate or hide or integrate its own DNA into the host DNA. You can see how it has hide this DNA, right? So now this DNA is hybrid. Okay, now this DNA will remain inactive and only will get active as and when required. So now once this needs to be activated, what's going to do? Now it is double-stranded DNA but integrated with viral genome, right? So now what's going to happen is, okay, now virus is going to use host cell machinery. Fine, host cell machinery. And this host cell machinery, with the help of this host cell machinery, this DNA, hybrid DNA, is going to get converted into, remember the central dogma, it's going to get converted into mRNA, which is going to get converted into proteins. Okay, and plus some RNAs as the genetic materials will also be synthesized. Okay, so now virus has used host cell machinery to produce all of this. Got it? Fine. Right. Now what will happen? Protein is formed, RNase is there. Can you imagine now the virus can assemble? Right? Okay. So now this protein is there. Fine. This is the viral protein. Remember. Okay. So this protein is going to be in form of long chain. So again some enzyme is required. So protease enzyme, which is the virus enzyme, is going to help them out. It's going to convert this protein into small fragments, and already RNAs are also there as the genetic material. So now can you imagine what's going to happen? Assembly of the virus. Yes, a virus is formed now. Okay, so now the virus will formed. Okay, assembly has been done. Now this virus needs to go outside, right, in order to infect other cells. So now it will push outside, right? Okay, now it will push outside. Fine, but while pushing, what's it going to do? It's again very smart. It's going to steal this outer portion of the host cell membrane, some part. Okay, some part of the host cell membrane is going to steal on its own, right? And it's going to incorporate for its structure. Now this part contains sugar plus protein, which forms glycoprotein. Now you can know, right, what is the function of glycoprotein? Right, it's used for binding to the another host cells, right? So now it has got glycoprotein also. So this structure will look like this. Finally, it is complete, right? With everything, even GP120 is formed. And now this cell will infect other cells, right? Okay, the work is done.